Hi everyone. Today we are going to discuss about Medicinal Chemistry Unit 1, in which we are going to discuss about physicochemical properties in relation to biological activity. What do you mean by these physicochemical properties? Why we need to consider these physicochemical properties? What is the reason behind that? So here, for a drug to show its activity, just having a good therapeutic action is not enough for that. We need to consider various parameters for a drug to show its response, because just having a good activity is not enough for a molecule to be effective. There are certain various properties of the drug has to be keep in consideration, so that when the drug is taken inside, it should elicit its action and our body should show that response. So we need to keep in mind about what are the various physicochemical properties and how they are going to affect the biological activity. So what do you mean by this physicochemical properties? So here, ability of chemical compound. So whatever the chemical compound we are having, any medicine or drug, its ability to elicit a pharmacological response is related to the influence of various physicochemical properties. Simply it doesn't show full response. There are various parameters which are involved and which have to be considered. If we are going to ignore these physicochemical properties, then our drug, even though it is showing a good results in our clinical trials, even in the initial trials, but if we are avoiding these physicochemical properties, then our drug won't be that much effective. So here, what are these physical properties? Physical property is a physical property of a drug is responsible for its action. Like whatever your drug is there, its physical property, whether it is a liquid, solid, and all that thing is responsible for its action. And chemical properties, because after taking that medication, our drug has to go through various biological reactions and all those things it has to be undergoing. So drug interact extracellularly according to simple reactions, neutralization, chelation, oxidation, and it is going to show its response. So all these parameters which are there, that has to be keep in mind. So now let us see what are these physicochemical properties. So various physicochemical properties are, first thing we need to keep in mind related to a drug is its solubility. Okay, solubility plays a very important role. We'll discuss few of them individually now. So these are the various physicochemical properties has to be kept in consideration while whenever we are being identifying a new drug or medication. We need to see that these physicochemical properties are justified, then only we will be selecting the best molecule. So solubility, we need to consider. Next, partition coefficient. Partition coefficient means what is the concentration of the drug in the lipid layer, what is the concentration in the water layer, like that sort of concentration reagent is called as partition coefficient. Ionization, what is the charge of the molecule, whether it is having a positive charge, negative charge, what is the actual how much amount of charge it is showing while binding the receptors, because there are different types of receptors present in our body. Fewer ion gated channels, fewer G protein coupled receptors, fewer sodium gated channels. So here, where our drug is going to act, we need to keep in mind about its ionization property. Next, hydrogen bonding, because drug is going to interact with the receptor either by binding to it or getting attached, maybe by through van der Waals forces and all. So hydrogen bonding also plays an important role here. Next is chelation. Okay, chelation is nothing but a formation of a complex with the drug molecule as well as the protein part or various receptor site. There might be some sort of chelation. So chelation has many effects, like even it could be giving side effects are also related to chelation. Even antidote property of a certain medication is based on the chelation mechanism. Next, surface activity is also needed for certain drugs to show good action. Then isosterism. Okay, so isosterism is nothing but having same number of electrons. If you are taking different molecules but having same number of electrons in that, then those molecules might elicit similar pharmacological response. So that are called as isosters. Okay, isomers, how we can tell that having the same chemical formula and different like showing this the same molecular formula and having the different structure, we call them as isomers. Isosters are those molecules being chemically different, but when they show same pharmacological response, those are called as isosters, and that is called as isosterism. To be specific, it is called as bioisosterism. Okay, so these are the various physicochemical properties that has to be considered for a drug to show a better biological activity in the body. So we have solubility, we have partition coefficient, ionization, hydrogen bonding, chelation, surface activity, isosterism. Now, let us see individually few of the physicochemical properties, how they are going to affect the biological activity, and how can we enhance molecules solubility or change the partition coefficient values. What we can do with that, let us discuss. So here, solubility is defined as the concentration of a dissolved solute which is equilibrium with the solid solute. Means when you are mixing two solutes with each other, how much amount of concentration of your added solute is with the equilibrium with the earlier present solute. So here, sufficient solubility of drug is mandatory or necessary for penetrating or membrane permeability. Because here what happened, when the drug has been taken inside, that has to be actually undergoing systemic circulation. It is going to inside, and then it is going to go intracellular, extracellular. It has to enter the cell wall and all. Different membrane has to be crossed there. So here, sufficient solubility is the main important part that has to be necessary. Like for oral absorption drugs also, they has to be have a certain amount of solubility needs to be kept in mind. So here, how we can identify the solubility? We can identify by buffer and ionic strength, by purity of the sample, polymorphism if it is showing. Then pH also impacts the solubility, supersaturation, thermodynamic and kinetic solubility are certain parameters which help to identify how much soluble the compound is. Now, if we go towards how this solubility is associated with other physicochemical properties, like what happened means as the solubility increases, then other physicochemical properties like boiling point, viscosity, surface activity, partition coefficient, if they increase, solubility decrease. If they decrease, solubility increase. These are like inversely proportional to each other. If you go towards a homologous series of a certain compounds like alcohols, and if you go towards different physicochemical properties like boiling point, viscosity, surface activity, if these things get increases, solubility can get decrease. So we are discussing like solubility, drug has to be soluble. Okay, that's fine. But how can we change or increase or decrease the solubility? So here, there has been given two drug examples in which by changing the salts, but by changing their respective salts, we can convert a water soluble into water insoluble, or water insoluble compound into water soluble compound. Example here given is methylprednisolone acetate. Methylprednisolone, it is a drug. Uh, and acetate is its salt. Okay, so we are taking methyl prednisolone acetate. This drug in this acetate form, it is water insoluble. It won't dissolve in water. But if we change that into methyl prednisolone sodium succinate, so you need to focus here. What we are changing here, we are changing that acetate into sodium succinate salt. Initially, it is an acetate salt. Now, that acetate salt has been modified into sodium succinate. So what is happening? Initially, when it was in acetate form, it was insoluble. Now, this acetate was water insoluble. Now, when it has been changed into sodium succinate, it has become water soluble. So, when we are changing their salts, we are improving the solubility here. Another example, if we go towards here, is of chloramphenicol. Chloramphenicol, it's usually used for eye infection. Chloramphenicol respules are used. You might have known that. So chloramphenicol, it is slightly soluble. Okay, so look here. Always it is not necessary that molecule has to be water soluble. It's not necessary for every drug. It is depending on where the drug has to dissolve, where the drug has to act when it is going towards whether it is going towards the stomach or whether it is going to release in the intestine, the solubility parameters changes. So here for chloramphenicol, we need lesser solubility in water, means lipid solubility has to be more. Whenever water insoluble means it will be lipid soluble. So here what happened, chloramphenicol is slightly insoluble, slightly soluble compound, and we are changing it to water insoluble or lipid soluble compound by changing it into palmitate salt. So chloramphenicol palmitate is insoluble in water or soluble in lipid more. Okay, so lipid solubility get enhanced for chloramphenicol when you are changing it into palmitate salts. So here you can see that how by changing their salts into their different forms, we are improvising the solubility. So solubility is a simple parameter, but it is very important here. If we keep in mind about where the drug has to release, where it has to show the action, which sort of membrane it has to cross, where the action is there, so we can select which is type of solubility, like water solubility is needed or lipid solubility is needed. So if we want to convert it to lipid solubility, how we can do, like we can change it into various salts, changing their salt form, we can enhance the solubility here. So now it has given how you can improvise the solubility of different compounds. So we can do that solubility enhancement by different methods. Like first, just now we have seen structural modification, modifying the structure. We are changing the moieties, salts, acetate to palmitate. We are changing, and that can be done. And another method is adding up of co-solvents. If you're adding a few more co-solvents to the along with the drug, then it also increases the solubility. Even employing surfactants helps in the solubility as well as complexation. Okay, so these are the four various methods that could be applied to enhance or improvise the solubility of the drug based on where the drug has to act, which type of solubility is preferred, we need to select. So it will vary from drug to drug, its dosage form also, the variation occurs. So solubility, if the drug solubility is very less, even though it is highly strong, very potent, then it was not soluble, it won't be showing the proper action. Now coming to next parameter, that is partition coefficient. Partition coefficient is nothing but amount of molecule, concentration of molecule in lipid layer and the water layer. The ratio of lipid and water concentration of a drug is called as partition coefficient. So in this, we will be seeing about both of the ionized molecule as well as unionized molecule. So we will be getting the degree of ionization here, it is A. So for ionized molecule concentration, how much it will be? The partition coefficient will be drug concentration in lipid layer by drug concentration water layer. For ionized one, concentration of drug in lipid, 1 minus A, that is degree of ionization in aqueous, and to the concentration of drug in the water. So why this partition coefficient is needed? This partition coefficient will be giving us the lipophilicity. Okay, so lipophilicity can be obtained through this partition coefficient. So here, factors which is going to affect the partition coefficient includes pH of the drug will be affecting the partition coefficient values, co-solvents, as well as complexation also can affect here. And partition coefficient are difficult to measure in the living system. That's what I have just said. So we will be considering the lipophilicity parameter. So here they are usually determined in vitro for that, because we cannot directly check in the human body. So we will be considering 1-octanol as liquid phase and phosphate buffer of 7.4 as aqueous phase. Then they will be comparing how much amount of drug is present in going in the octanol layer and how much is going in the phosphate buffer layer. With that, we will be knowing the partition coefficient value of the drug in vitro. Like in laboratory itself, we can identify, okay, this much amount is going in the octanol layer and this much is going in the phosphate buffer layer. So this will be the partition coefficient value for the drug in the body, in aqueous layer as well as in the lipid layer. So here, partition coefficient, in that when we are adding up log, just what I have said, lipophilicity will be defined through this partition coefficient. So when we add the log P will be the measure of lipophilicity, how much amount of drug is able to dissolve in the lipid layer. Lipophilicity of the drug will be identified through this P values. So here we are having various methods through which this lipophilicity can be identified. We have shake flask method, chromatographic method, and spectroscopic method. Okay, so these are the various methods by which we'll be getting the log P value. So how this partition coefficient will be effective for the drug? Let us take an example and then identify. So here we are taking phenobarbital and we are comparing it with thiopentone sodium. Okay, so both are belong to category of sedative and hypnotics category. So if we go towards phenobarbital, it is having lipid water partition coefficient of 5.9. But if we take thiopentone sodium, it will be having chloroform water. Again, it will be lipid water coefficient of about 100. So we can see that where is 5.9, where is 100. So we can select which would be the most effective when compared to that phenobarbital. Thiopentone will be having better effects. The reason behind it's not about its effectiveness towards the receptors and all. It is based on its partition coefficient. It is having high solubility in the lipid layer. It will be penetrating well and it will be going towards the receptors, and will be showing better action because it is having a very high partition coefficient, due to which solubility in lipid increases and it can cross various lipomembranes layers inside the body. So here, that is why this partition coefficient value plays a very important role to predict various derivatives of the drugs are there. So which will be the highly efficient, which will be very strong. This we can identify through the values of partition coefficient. We can predict that, okay, this is having very high value. So it will be having high penetration layer. So these can be given at very low doses also. They might be able to go inside and they might be going to show their effect because it is having so high value for partition coefficient. Now, surface activity. So why the surface activity? So here, this is also an important parameter, surface activity, because surface activation agent will be affecting the drug absorption. Like when you're adding the because in the property of solubility itself, we have seen that surfactants can enhance the solubility. So sometimes, in order to improvise or increase the solubility of the compound, we will be adding these surfactants which will enhance the solubility. So, drug absorption, how it will vary because based on the chemical nature of surfactant, how it is, which sort of molecule you need to select. Its concentration also plays an important role, because if we go towards surfactants, they have been having a property of micelle formation, and that will be helping in penetrating the molecules inside. We know that in the cleaning action of soap, how these surface active agents are responsible to trap the dirt inside. So instead of that, it can also trap a drug material inside, and it could enhance the solubility. The same thing. Instead of, you can just imagine how the surface active agents, after forming micelle, they will trap the dirt into it, and it will go inside the water. The same property can be done with that of drugs also. These micelles, they can trap the drug molecule, and it can take it inside the various membranes and increases the solubility. So here you can see how these surface active agents are responsible in various different categories. So you can see here their applications. At lower concentration, surfactant enhance absorption rate. At higher, it will be reducing the absorption. So very high also is not needed, but when they are being given at optimum values, they will be enhancing the absorption rate. So you can see here, anti-helminthic activity of hexyl resorcinol is due to this surface activity. Bactericidal activity of cationic quaternary ammonium compounds is due to this surface activity. Bactericidal means mostly this topical antifungal, antibacterial agents which are there. They show their activity through this surfactant surface activity nature. So this disinfectant action of phenol and cresol is also due to this property. So now coming to another very important, very important physicochemical parameter, that is hydrogen bonding. What is hydrogen bonding? You might be knowing that. Hydrogen bonding is, by the name itself, there is a bonding of hydrogen. Hydrogen is creating a bond. So this is not a simple bond. It is an extra, special type of interaction that is happening when there is hydrogen and a very highly electronegative element, okay, electronegative element, such as nitrogen, sulfur, and oxygen. When they are there in a molecule along with the hydrogen, what happens? Hydrogen creates a dipole-dipole interaction with that, due to which what happened? Extra attachment will get increased. So these weak bonds, usually hydrogen bonds, are denoted by the dotted line. So here what is happening? Actually, hydrogen was attached to OH here, but it is having another oxygen nearby. So there is a, due to that high electronegativity of oxygen, it is having an interaction with the hydrogen. Similarly, with hydrogen and nitrogen also can have this sort of dipole-dipole interaction. So here, the compound capable of forming hydrogen bonding is only soluble in water. So water itself has this hydrogen bonding. The existence of, you might be knowing that, H2O and H2S, this two molecules, if you take water and H2S. Actually, oxygen and sulfur both belong to 16 group element. Both will have similar property, but this is liquid and this is gas. Reason that hydrogen bonding is seen in water. dipole-dipole interaction will create in water molecules, and it will be having closing that molecules into a liquid state. Whereas in H2S, there is no hydrogen bonding, so it is in the gas state. So similarly, this is some sort of property of hydrogen bonding, but this hydrogen bonding is also important for our biological activity. Biological activity of the compounds get affected if there is hydrogen bonding. Sometimes if it is hydrogen bonding is not there, then also it is get affected. So let us see a few examples and understand. So here, this hydrogen bonding, it could be of various types. Two major types of hydrogen bonding are there. So one type is intermolecular. Okay, intermolecular hydrogen bonding. So what do you mean by intermolecular? When the bonding is between the molecules. I'll give an example here. Water. This is the structure for water. This is one water molecule. There is another water molecule. So here, these are two different water molecules, but this hydrogen will create a hydrogen bonding with this oxygen. Similarly, if there is another, like there are many of water molecules will be there in a single animal, thousands of water molecules will be there. So what happens? Similarly, all of them, they will be having this sort of interaction with their nearby elements. And due to which what happened? Water is holding all together and creating that liquid state. Okay. So this is inter. Inter means in between the molecules. So this is one type of hydrogen bonding. Interhydrogen, intermolecular H bonding. So you can see that in HF, hydrogen fluoride also, similar sort of hydrogen bonding is being seen. In that also, it will be getting attached with another molecule. Even hydrogen fluoride is in liquid state due to this hydrogen bonding effect. So this hydrogen bonding is very important for molecules to have a specific type of state. So similarly, there is another type of hydrogen bonding that is intra. Intra means when single molecule in that itself there is an interaction. Okay, that is called as intramolecular hydrogen bonding. So if we go towards intra, we go towards intramolecular. Intra means within the molecule. So here for this we are taking example for nitrobenzaldehyde. Okay, so orthonitrobenzaldehyde. So here this is a benzaldehyde molecule having this H and a nitro compound. NO will be there here. NO2. So here, this oxygen of NO2 will be having hydrogen bonding with the aldehyde. Okay, so this sort of hydrogen bonding is happening within the same molecule. So this is called as intramolecular hydrogen bonding. So this is another type, that is intramolecular hydrogen bonding, where the bonding is happening within the same molecule. There is a dipole-dipole interaction that is happening in the molecule. How this is effective for a drug? So here, this intramolecular, and to be specific, this intramolecular hydrogen bonding is highly effective for salicylic acid. Okay, so here, if you take salicylic acid, salicylic acid is nothing but COOH with the hydroxyl group on ortho position. So this salicylic acid, if you take among these different hydroxy derivatives of carboxylic acid, only salicylic acid will be having the high effectiveness. Why? Because of the hydrogen bonding. So here, intramolecular hydrogen bonding is needed for salicylic acid. If I take another derivative, this is O-hydroxy. Instead of O-hydroxy, if I take para-hydroxy, that won't be effective. If hydroxyl group is at para position, it won't show much effect. It is also having COH here. This is para-hydroxybenzoic acid, and this is O-hydroxybenzoic acid. And even if you have meta-hydroxy, this is para-hydroxy. So this won't be effective because this sort of here, this much distance, it cannot travel for hydrogen bonding. But here, this is effective. This is our salicylic acid. So salicylic acid is O-hydroxy, orthohydroxy benzoic acid. Why this is only effective? Because it can show H bond. Hydrogen bonding is seen in this. So similarly, hydrogen bonding is sometimes it is mandatory, necessary. If it is not happening, then it will like same molecular formula, both are having hydroxyl, but ortho one is effective. Para, even meta is also not effective because there won't be any hydrogen bonding in that. So this way, hydrogen bonding is also necessary. Like always, hydrogen bonding should be there. It's not needed, but certain things, certain molecules, hydrogen bonding is important for the drug to be effective. One of the example for that is salicylic acid. Now coming to another physicochemical property, that is chelation. So what do you mean by chelation? Like chelation is nothing but by donating electron to the metal, it form a ring structure. Molecule will form a ring structure that is called as chelates. So the molecules which are forming that ring structure, these are formed by bonds forming the ring that are termed as ligands. Which type of bond? The molecules which are forming all together and creating that ring, those bonds capable of that are called as ligands. So ligands are needed for a molecule to form the chelates. So here, chelation is highly effective and it is needed for showing the activity here. So here we have examples for 8-hydroxyquinoline and its analogs. These are antibacterial, antifungal property that are there, and that is happen to be formation of complex with iron or copper. Okay, so this chelation, formation of chelates with iron and copper by 8-hydroxyquinoline is due to this chelation. And there are certain undesirable side effects can also be happen with the drugs. For that, we have example for metals like hydralazine, an antihypertensive agent, is formation of anemia occurs with that, one of its side effect. And this is happening due to chelation of drug with iron. Here, what is happening? It is forming a chelate with iron, due to which it is showing that property of anemia. Its side effect of anemia can be done happened due to the chelation with drug with iron. So this hydralazine is now being not used because of this anemia sort of side effects it might be giving, because it is what is doing is creating a chelate with iron and causing anemia, decrease the hemoglobin level in the body. Okay, and apart from that, chelates are also very much important for trapping of poisonous compounds. Okay, so like if we take arsenic, arsenic poisoning, if you're having, okay, so this arsenic poisoning can be eliminated by the complexing with the various metals. Okay, so with cisplatin or such metals, this arsenic will get eliminated through chelation. So various antidotes for poisoning are being done through this chelation complexation property. So dimer is also an example for chelate antidote, which is used for various poisoning, metal poisoning. We will be using this dimer, which also forms chelates, and it will be removing the various poisonous metals from the body. So chelation is sometimes beneficial. It is having various activities, antidote property, but sometimes it is also having undesirable effects. It will be giving undesirable effects. Like one example we have seen about hydralazine, where it will be causing anemia because it is creating a chelate with that of iron. So here, what we have discussed till now, these concepts are very important for your exam point of view. I think you might be having a quiz session in the upcoming this YouTube message box, in that they will be sending a link in which you will be having multiple choice questions that will be asked related to our session webinar today. You can give an answers related to that, which will be very beneficial for you. Okay, so these physicochemical properties are very important for exam point of view also, because they have been asked for 2 marks, 5 marks, 2, 3 marks, even in very short also they have been asked. So you can see here hydrogen bonding solubility togetherly it has been asked sometimes. Sometimes chelation and bioisosterism. We have not discussed now because time bond is there. So bioisosterism chelation, that has been asked in our previous question papers. Protein binding and geometrical isomerism. When this bioisosterism concept get discussed, at that time this geometrical isomerism also might also be considered at that time. So these are also various physical-chemical properties. So they will be asked 2 or 3 together in their exams, in which you need to write about that. And one important point you need to keep in mind whenever you're going to write about physical-chemical properties, as we are discussing these physical-chemical properties relating to drug activity. So give a drug example for justifying your physical chemical property. We have seen solubility, we have seen drug examples, partition coefficient, we have seen drug examples. So whenever you're with hydrogen bonding, we have seen drug examples. So likewise in chelation, we have seen drug examples. So whenever you're going to write about this physicochemical properties in your exams, you should be giving an example of a medication, how these physicochemical properties are improvised as a medicines, and how we can get new, better medication by doing what sort of addition into that. And we need to show like how, like just now we have seen, salicylic acid property is due to the presence of intramolecular hydrogen bonding in that. So likewise, solubility, we have taken methylprednisolone, example, acetate format, if it is changed to sodium palmitate, it will be having a more solubility. Okay, so sodium succinate, sorry, methylprednisolone, sodium succinate is having higher improvising the solubility part. So in that way, for each category, whenever you're going to study that, then you should be giving a drug example with that. So apart from this, in this first unit, you're having topics such as prodrug. This is also an important concept which you need to focus on, and drug metabolism, phase one, phase two reaction. For OU syllabus, this drug metabolism is there. For JNTU students, they have not included the drug metabolism now. They have removed that part. But for OU and PCI syllabus, we are having this drug metabolism, in which we need to discuss about phase one, phase two reactions that are there. So that's it for today's session. And for further, if you want to have a more elaborative study related to medicinal chemistry, various units and all, you can follow the app. You can subscribe to the YouTube channel of Prakara, from which you have been joining through YouTube. You can subscribe to the channel. You can follow us on Instagram, and you can download our app and have a full elaborative sessions you will be having, where you can buy the various modules in which you can go through in-depth, full detail we can discuss. So that's it for today's session.